Welcome back, Biology 202. Today we're going to be discussing the immune and lymphatic systems. So just some terms to get started. We define immunity as the body's resistance to disease. So how resilient are we to disease and infection? The immune system is described as a functional system in the sense that it's trillions of individual cells, and these cells are scattered throughout our body. And the functionality of that, there are several things we're going to look at, but the biggest things will be, of course, identifying invaders, providing a specific attack for specific invaders, and then being able to remember past invaders so that the next time you encounter those same invaders or something very similar, your body is able to fight them off more readily. So the immune defense system types, we have two major types, and we'll look at some different layers or lines of defenses, but we have what's called the innate and the adaptive. If you took microbiology, you've probably, almost all of this PowerPoint you have already seen before, basically all the basic things in it. But for innate immunity, we say this is non-specific. It responds within minutes, and this would be against all foreign substances. So our innate immunity is what we're born with. Versus adaptive immunity, that's where we specifically respond to particular invaders, and we acquire this over time. Now, some different ways to look at innate and adaptive immunity. For innate defenses, we have our first and second line of defenses in this category. A first line of defense would be some type of surface barrier. So for that would be your skin, right? You're born with your skin. It has an inhospitable environment for certain pathogens. And it's just something you're born with, and it's this surface barrier. And that's going to prevent bacteria from landing on your skin and then just going right into your vascular tissue and causing sepsis. So you have this nice wall there to prevent infection. Mucous membranes also would be considered a first line of defense. It's the first thing an invader is going to run into in your body. They're going to be on your skin, or they're going to be breathed in and interface with a mucous membrane. So that is a barrier that prevents that bacteria from just going right into your body and infecting you. Our second line of defense, these are also innate defenses, would be examples on the inside of our body, such as phagocytes. So white blood cells that can grab hold of a foreign substance and phagocytize it or break it down into smaller pieces. A fever is something that's a pathway that we'll have that will increase the temperature and make the temperature outside that ideal range for the bacteria. Natural killer cells, antimicrobial proteins, and the inflammation pathway. This is going to be the second step that a microbe is going to run into, or a foreign material, when it infects our body. Now, if a bacteria is able to get through our first line of defense and our second line of defense, then they're going to run into our third line of defense, which we call adaptive immunity. And we acquire this as we live our lives and as we are exposed to different foreign substances. And we'll look at this in more detail, but just to throw these categories out at you, we have humoral immunity. This is going to involve B cells, and then we have cellular immunity, which is going to involve T cells. So let's look at the innate defenses, these non-specific defenses. So, of course, we mentioned the surface barrier. So this is the body's first line of defense. It's going to be your skin and mucous membrane. Anything that lines a external opening. So anywhere where there's a barrier between the inside of your body and the outside of your body. So these are general protection. They're not necessarily designed for certain things. They just block everything. It's a physical barrier. So you have layers of cells. You have fluids and antimicrobial chemicals and proteins and enzymes that are released into these mucous membranes. They're going to help fight off bacteria, destroy them, and then flush them out. So this keeps bacteria from getting into your body. And then generally the pH and things of that sort, the chemical nature of it is going to be inhospitable for microbes to survive. So how is this environment going to be inhospitable? Well, for the skin, there are certain secretions that help get the ideal range of the pH and salinity away from what microbes would normally prefer. So we see that with our sweat and sebum, the fluids and oils that build up on our skin. They create a more acidic environment that is inhospitable to the bacteria. We also have cerumen, which is earwax, and this is going to come from the ceruminous glands in the ear canal, and that's going to contain toxic chemicals towards bacteria. Think about vaginal secretions; those are also highly acidic. Our stomach drops to a sub 2 pH, which obviously is going to kill a lot of microbes. The stomach isn't really about breaking the food into smaller pieces as much as it is basically covering the food, if you've chewed it well, hopefully, in acid to kill any microbes so that they don't move farther down your digestive system. Saliva and tears. These are filled with lysozymes that flush the bacteria not only off your eyes, but also kill the bacteria that is there. And then, of course, we have this mucus that helps trap bacteria so that we can destroy them with phagocytes and lysozymes. So these general areas, just creating an inhospitable chemical environment is going to prevent so many infections that would otherwise potentially kill us. So it's great that we have these natural mechanisms of defense. So then internally, we have these internal innate defenses. And for them, they're able to identify the pathogen by some recognition that something is different. So they recognize things on the surface of those cells of microbes to be able to tell that they are not self, and then they're able to destroy them. So this would be cellular and chemical defenses. And these provide the body's second line. This is what's going to happen after the bacteria maybe gets through the skin or through a mucous membrane. And we'll have these phagocytes that can eat that foreign material. So some examples of phagocytes, we got macrophages. We consider these, we think macro. So that's going to be large. These are large eating cells that come from monocytes. And they can either be wandering, where they roam around, or they can be fixed in a particular organ. We also have neutrophils. These are going to be the most abundant white blood cell in our body. If you remember, we said these were the standing army in the body, considering as far as our white blood cells go. When we were looking through those blood samples, you're probably going to find a neutrophil before anything else because they make up to almost 2/3 of the white blood cells. So it's that standing army. They produce antibiotic-like chemicals. We call those defensins that can help destroy these bacteria. And other chemicals they'll release that harm bacteria. So they're just sitting around and they're ready to go to fight off these microbes when they run into them. These numbers will increase during certain infections, for example, appendicitis, meningitis. If your white blood cell counts are increasing, then there's a chance of infection there. Could indicate that there is an infection. Eosinophils, weakly phagocytic, and they defense against certain parasites they work well against. So for white blood cells to move towards the injury site, well, they're going to be flowing through the vascular tissue anyway. They're going to be made in the bone marrow. And then they're flowing through the vascular tissue. So when they come to an injury site, they'll start to bind to the endothelium, and they call that process margination. Then when they leap through the lining of the vascular tissue, they call that diapedesis. Dia in Greek means through. So pedesis, if I remember, is like leaping. So it's literally leaping through the vascular tissue. That becomes more permeable through the inflammation pathway. And then the white blood cells, the phagocytes, can then be attracted to the chemicals that are being given off at that injury site. We call that chemotaxis, chemical movement towards the chemicals if it's positive. And then they're going to be able to phagocytize the microbes. So what are some ways that they can do this? What is the mechanisms of phagocytosis? So first, they have to be able to stick to it. So we call that adherence. So the flowing cytoplasmic extensions of the phagocyte bind to the foreign particle. And I'll show you all some pictures of that. It looks pretty neat. Engulfing, so the pseudopods engulf the foreign particle. So endocytosis is when a cell brings in something into the cell process, endocytosis. Phagosome formation, so the foreign particle is pulled inside the phagocyte and enclosed within a membrane vacuole, creating a phagosome. And then phagolysosome formation, the phagosome fuses with a lysosome, which has the digestive enzymes, so that it can actually break it down. And then the waste products that's released, we call that exocytosis, the movement out of the cell. And this is just a diagram of what that looks like. So the microbe touches the cell, then the pseudopods, like an amoeba, are going to wrap around it. It's going to bring it in a vesicle. So you see how the membrane ties to it, and it has its own little membrane that it's enclosed in, so it's compartmentalized. Then it will combine with a lysosome that you see here with these digestive enzymes, and they're going to mix. Digestive enzymes break it down, literally digest it, and then spits it out, exocytosis. And here is an image. You can actually see the bacterial cells all around this large white blood cell, and it is bringing them in and digesting those microbes, which is pretty cool. That is a real picture of what that looks like with electron micrography. And then there's another image of two that are more closely inside, in this case, inside a lymphocyte attacking a cancer cell. So that's why it's so large, what it's consuming. It's pretty interesting. So another type of cell we're going to have is called a natural killer cell, often abbreviated an NK cell. So these cells, they're large, granular, nonspecific, non-phagocytic lymphocytes. And these are usually described as the patrol, like the police in the body. And they're in the blood and the lymph, and they can kill these cells directly. That's why they call them natural killer cells. So they'll often be called the pit bulls. They're roaming around, ready to kill these foreign substances. And the way they do that is, you see, it looks like they got a bunch of guns sticking out of their body over here. But what they do is they can target a cell's membrane, and then they release chemicals into it that create these holes, and then it causes it to self-destruct. So it's a very quick, not necessarily specific defense system. It can just find a foreign substance and destroy it immediately. So it's a very fast way to get rid of a foreign substance. Natural killer cells. So that brings us to the inflammatory response. So we've all had inflammation happen before. We probably remember some, intense injuries we've had or things where this has taken place. But the definition of it is a localized response triggered whenever body tissues are injured due to physical trauma, intense heat, irritating chemicals, or infection by viruses, bacteria, and fungi. And the purpose of it, it's not just that there was damage occurred and this is just like the result of that, like bruising or something. This is a part of your body, a system that is designed to help fight off some of the secondary things that happen due to physical damage. So some functions: number 1, prevents the spread of damaging agents. Number 2, disposes of cell debris and pathogens. Number 3, sets the stage for repair processes by bringing nutrients and workers, such as white blood cells. And then the 4 signs of inflammation would be redness, heat, swelling, and pain. All of these have a purpose. If we're talking about an infection that you might have from bacteria, for example, the swelling that occurs is purposeful, and the heat, it can get the bacteria away from their ideal temperature, which can make it harder for them to replicate, which gives your immune system more time to identify the pathogens to destroy them. The swelling, for that, what's happening is there's increased blood flow to that area, which brings more immune cells, more white blood cells, so that they can target those bacteria for destruction and release toxins on them, which can cause more heat. It can cause more redness and pain. And then it will start to facilitate the healing process, which more blood flow helps with that as well. So it's all basically like, let's bring some attention to this location where damage has happened, kill any bacteria that are there. If not, then we need the increased blood flow and swelling for repair purposes anyway, so then it can jumpstart that process. So it's to increase the rate of fighting off the infection and healing your body. So it's not meaningless. So these are the events that can take place to allow those 4 cardinal signs to occur. So number 1, inflammatory chemicals are going to be released. It's going to be histamine, prostaglandins. That's going to come into the extracellular fluid by the tissues that were harmed themselves. We're going to have phagocytes, lymphocytes, mast cells, and blood proteins. Then number 2, the chemicals will cause small blood vessels in the vicinity to dilate, which is going to bring in more blood flow, which causes that natural redness and temperature increase that you see at the site of an injury. The chemicals will make the permeability of the vascular tissue increase, so we can have that diapedesis of those white blood cells moving from the vascular tissue into the area of injury. So then what can happen, or what will happen, is the exudates, which means the fluid that contains the proteins that will be important for fighting off the infection and stopping blood loss or anything like that, such as clotting factors, antibodies. Those are going to seep from the bloodstream into the tissue spaces, and that can cause that edema. And edema just means swelling. There can be other causes for it, but this could be one of the causes. And when you have swelling, that can press on nerve endings, which can stimulate it, cause an action potential, and then it sends that signal back to your brain, and you feel pain. Then the injured area is invaded by more phagocytes. First, your neutrophils, that's the standing army. You're going to have the macrophages moving in. Leukocytosis will take place, so you're going to make more white blood cells in your bone marrow so that you can have more available to fight off that infection. And then that would show up in a blood test. That can be a sign of an infection somewhere in your body. You'll have margination or pavementing, where they bind to that endothelium. Diapedesis is where they move through it. And then chemotaxis is where they're attracted by those inflammatory chemicals. So here's some different things that can be working together that can all be happening to allow for the healing, ultimately for the healing of an injury. So we'd start up here at the top. You have the injury. And let's say we follow this pathway. Well, we're going to have more white blood cells produced. So we're going to end up with leukocytosis. Those white blood cells are going to migrate to the injured area. They're going to move through the vascular tissue. Diapedesis, after they bind to the endothelium. Diapedesis, leap through the endothelium. And then you're going to have phagocytosis of the pathogens. And then you might get some pus, which can be a collection of fluids and dead bacteria piling up. And then you have to clear the debris and repair, and you'll end up with healing in the end. We have antimicrobial proteins that'll play a role in fighting off that infection. We have something called an interferon, and they're named that because they interfere with the ability of a virus to reproduce. They're going to be made by different leukocytes, such as like monocytes, macrophages, B and T cells, and complement proteins. These amplify the inflammatory response, and it can destroy the pathogens, and it can mark the bad guys for destruction that they call opsonization. And those can be made, primarily they're going to be made by the liver, certain liver cells called hepatocytes. You'll see it made by macrophages, monocytes, and different things like that. So there's a little bit of an overlap in what can make complement proteins and interferon. So what is a fever and why do they occur? Well, a fever is defined as an abnormally high body temperature. And anytime our body changes the environment, whether by its nature, like the stomach having a low pH, or a fever, inflammatory response, things are changing quickly, usually part of that reason, and not always the only reason, but part of that reason is changing the conditions outside of the comfort zone, we could say that, of the bacteria and their enzymes that those bacteria have. So for us, this abnormally high body temperature can get away from the ideal temperature of the bacteria. It's a systemic response initiated by pyrogenes, so secreted by leukocytes and macrophages that are exposed to these pathogens. And then it's denaturing enzymes, so it can destroy the enzymes the bacteria are using, but it can also harm our own bodies because it can get such a high temperature that our own enzymes start to denature. The liver and spleen sequester iron and zinc because those are needed by bacteria to survive. So our body pulls any of that out of the way. And then the higher temperature and these other factors are going to cause it to be very difficult for the foreign pathogen to survive in our bodies. And it's a good thing. A fever can be a good thing, but prolonged and too high of a temperature can damage our bodies. And especially in children, it can cause brain damage if you're at too high of a temperature for too long of a period of time. And that's one of the big pushes for vaccines, is that if you can prevent, maybe they'll still get infected, but because they have this built up immunity now, they won't have as high of a fever, or they might not have a fever at all. And even if they get infected with that pathogen, it's not going to be as hard on their bodies. And that brings us to our adaptive defenses. We call this the third line of defense. Now, we say that because it's the third thing, but it's going to be happening at the same time as the second line of defense. There is an overlap there. So it recognizes something as foreign and acts to immobilize, neutralize, or remove it. And it is specific. So the antigen that is presented, the foreign particle, our bodies make antibodies that can attach to those antigens so they can be targeted for destruction. Because we make specific antibodies for specific antigens, our bodies can remember through memory cells the antibodies that were made to fight off a previous infection. So it has memory, and this system is going to work so the next time we're infected, our response is stronger and quicker. So that's where we see it in this pathway. You're going to really have this third line and the second line happening at the same time, right? You're going to have inflammation, and you're also going to be making antibodies to target the microbes that are in that area that has injury and infection. And just to look back at the difference in antigen and antibody, antigens are the foreign particle. Antibodies is a protein, immunoglobulins, that we make to connect to the antigens so that they can be targeted for destruction. So antigens, antibodies allow us to target antigens. And the term antigen actually means antibody generating. So when that gets into our bodies, it generates or causes us to make these antibodies. So one of the obviously most important things that'll come up when you start talking about the third line of defenses and where we have this is going to be with the B cells and the T cells. So first, just the name, the reason they have these names is they're going to be made in the bone marrow, but B cells mature in the bone. T cells mature in the thymus. So T for thymus, T cells, B for bone, B cells. And what they're going to do is they're going to have different roles. The B cells, they attack invaders outside of the cells, and T cells are going to attack infected cells. And we'll look at how they do this. So for our primary immune response, the B cells are going to encounter the antigen for the first time, and they're going to have to do something to target them for destruction. The way they're going to do that is they're going to make antibodies. So first, though, the B cells will form what's called plasma cells and memory B cells. The plasma cells are going to make the antibodies that are able to target the antigen for destruction. During this whole time, you have the inflammation pathway happening. Temperatures are rising, swelling is occurring. The antibody will attach the antigen and allow it to be targeted so that it can be destroyed. The second time, so that's what you see on this first pathway right here in the graph. So we see here we have the naive B cell. It's going to have activated B cells, and they are going to produce these antibodies that target the antigens, foreign pathogen, or whatever we're infected with, for destruction. And then you'll have some of those plasma cells continue. You'll have a lower level of those antibodies in your blood. But there will still be some of those antibodies in the blood, right? You see, you start here, and then after you go through that first infection, you still have some floating around. So, like when COVID was happening, you could take an antibody test to see if you had been infected, to see if you had built up immunity, because you're still going to have some antibodies produced in your body. Now, the second time you're infected, what was also made, you had plasma cells and you had these memory cells. And these memory B cells on the second infection are ready to go, and they're rapidly going to produce plasma cells that rapidly produce higher numbers. Look at the total number of antibodies here to target more of those antigens at once for a faster destruction of that foreign particle. So on the secondary antibody response is going to be faster, it's going to be more efficient, and you're not going to be sick as long, and you're not going to have as many symptoms because your body gets rid of that antigen much faster because it's prepared for it. So one of the best ways to build up immunity to something is, of course, vaccination sort of takes care of this first part here in some sense. And we could talk about that more at another time. But it's basically getting the immunity from this process without all the side effects of a live pathogen. So then the next time you're infected, you already have these memory B cells that can make plasma cells and produce antibodies to target it more quickly. So here's another way at looking at humoral immunity. The word humoral, if we think of humor, or they used to have the four humor theory of health and sickness, which is incorrect, but humor just means a body fluid. So these are secreted into the fluids of the body, these antibodies, so that they can be targeted for destruction. But of course, you have that initial exposure to an antigen. It's going to be expose these initial white blood cells to these specific B lymphocytes, and then we're going to get our activated B cells that are then going to produce plasma cells that then make those antibodies to target the antigen so that it can be selected for destruction. And then every time that happens, memory cells are made so that the next time, here in the secondary infection, you have these memory cells ready to go to produce more plasma cells, to produce more antibodies, so on and so forth. So you get that infection off more quickly the second time. So what are the types of humoral immunity? We have active humoral immunity and passive humoral immunity. So active would mean you actively made or making the antibodies. And then passive humoral immunity would be you did not make these antibodies. So here's a cool way to sort of break it down. So let's say humoral immunity at the top. If we break it down into active and passive, for active, well, that could be naturally acquired. So you were infected, and then you made the plasma cells and the memory cells for that pathogen, and now the next time you naturally acquired a way to fight it off because you were actually exposed to that pathogen. So you had contact with the pathogen. Well, it could be active, but it was artificially acquired rather than naturally acquired. And for that, you might have been given a vaccine or a dead or weakened form of that pathogen. So it was artificial in that sense. It wasn't a natural exposure. It was artificially forced on you, but it was still active that now your body was tested basically to make these antibodies itself. So you still made the antibodies. You were just given a good, safe opportunity to develop your own antibodies. And that's what we'd see with like vaccination. Now, passive, we also have natural and artificial. So a passive humoral immunity that would be natural, example of that would be when antibodies are passed from the mother to the fetus through the placenta. So the baby didn't make its own antibodies. It was given them, so it passively received them, but it was through a natural means. This was a natural process that happened. Well, they could passively receive humoral immunity artificially if they're injected with those antibodies. And for certain infections, like for example, even with COVID, they had monoclonal antibodies that they would inject into people that would then give them the ability to target those COVID and the spike protein for destruction. So that would have been artificial. So here's just another way to see those types of immunity, and you will be asked questions about that. And there's just some images to kind of put in your head on what that looks like. So to kind of go into this for the details, so for the active humoral immunity, for natural, so you could have your B cells are making these antibodies because you were actually infected with an actual natural exposure to the pathogen for the infection. And then artificially acquired would be they gave you something that was weakened, but you still made your own antibodies. It was just from exposure to a weakened form of something that wouldn't be a natural encounter. And then for the passive humoral immunity, for naturally acquired, again, the mother passes it to her child through the placenta. So they didn't make their own antibodies, they received them passively. And then artificially would be they were given a shot of those antibodies, and here they have the monoclonal antibodies. So when the antigen comes into contact with the antibody, we form the antigen-antibody complex. And that can inactivate the antigen through several means. So it can neutralize it. So it covers it. It masks the parts of that organism that could harm us. So it covers it. We call that neutralization. It could do agglutination. So it binds to them and sticks them all together, which can make it easier for a phagocyte to come in and eat a clump of these bacterial cells. You could have precipitation. So in that sense, if the antigen is soluble, that means it's dissolved in a liquid. So for precipitation to happen, it makes it precipitate out. When I used to teach AP chemistry, we had some reactions where you mix two liquids, and then at the bottom of the test tube, you get a solid pebble that forms, or flakes that form. And precipitate, it's where you see the solid. You can think of it that way. So it precipitates out. All those things just make it easier to find it and destroy it. And then the complement proteins, like we saw, that's going to make it where they can be lysed and also phagocytized. So now we can talked about B cells. So let's talk about T cells. We got two major types of T cells. We have helper T cells and cytotoxic T cells. So for helper T cells, you can think of them as coordinators or helpers. And what a helper would do is it helps other cells do their job better. And helper T cells are going to coordinate or stimulate other immune cells, such as the B cells, like we just talked about, or the cytotoxic T cells, like we'll look at right here, by releasing a chemical, several chemicals, but we call the family of chemicals they release cytokines. The cytotoxic T cells, think about cytotoxic, cell toxic. These are the killers. They're toxic to the foreign substance. So they're going to directly destroy them. They can even destroy tumor cells by telling them to go into apoptosis. So you'll see some other terms for these, like helper T cells will be called CD4 plus cells, and the cytotoxic T cells will be called CD8 plus T cells. And the name CD4, CD8, those are named after a certain cell surface protein on the surface of their cells. That's where those names come from. So these T cells in a lymph node would become activated. They would multiply. And you could think of them because they're specifically going to go out and kill cells. Then we consider this like the army. So they're flowing out of the lymph and they're looking to hunt and destroy some specific threat. And you see these surface protein receptors that are sticking out of them, and they're ready to go find some foreign cells to destroy. So there are these cells called dendritic cells that are actually going to take pieces of the infection microbe, for example, these antigens, and they show them to the T cells. And if the T cell has a receptor that matches up with that antigen, then it's activated and it can go out to kill it. Now, the dendritic cell is shown here in the purple. It has all these little legs, which is why the name dendritic comes from. Dendrites are like branching, and they have all these little branchings, tree-like shapes coming out of the bottom, like you would see on tree branches. So they're basically like a spy. They're always patrolling, looking for foreign invaders, and if they find one, and this might even be from a cancer cell, which starts to mutate and look different than regular cells. It can engulf that cancer cell or engulf that bacteria, break it into tiny pieces, and then do what it's doing right here. It'll build up in the lymph nodes. It'll get there, and then it will present these antigens to those T cells. So when cytotoxic T cells encounter the antigens, then they are going to kill them. And it has to do with who they are targeting. So it's specific to a specific antigen. And then they have these little bows and arrows here. It shows they are designed to kill these specific bacteria. And it really is like a war going on, strategizing. You have these spies. They are helping to train these soldiers to describe, like, this is what you're looking for. Let's go destroy them. And then battles take place. And then somebody wins. And thankfully, our immune system wins all the time. And we are encountering these pathogens constantly. And we're pretty much constantly winning, which is the good news. But as we age, this will become less and less efficient. When the antibodies bind to these antigens, this can cause several different ways to destroy the bacteria, if we're talking about bacteria. So it could be antibody-dependent cell-mediated cytotoxicity. So it targets the antigen, and then a natural killer cell can find it very quickly and destroy it. Or it could be just targets it for phagocytosis. And then you have complement-dependent cytotoxicity. And basically what happens there is you have a complement cascade of events that takes place. And at the end of that, you get something called a membrane attack complex, or a MAC, that's going to punch holes in that cell. And it's going to result in it lysing and being destroyed. So when these lymphocytes are made in the red bone marrow, well, if they become a T cell, then they will mature in the thymus. So that's what they're showing you here in the middle, maturing in the thymus. Well, they could be helper cells, or they could be cytotoxic T cells. And to be activated, an antigen-presenting cell will present the antigen to them, which activates them. And they could form memory cells from that. And this is in the lymphoid tissue in different organs of our body. And they can replicate, and then you get these helper T cells in the blood plasma going out to help regulate this process. And then if it's CD8 cells that are activated by antigen-presenting cells, or what we call dendritic cells, you can see it looks like the dendrites that you see on a neuron. That's why they're called dendritic cells. And then in the lymphoid tissue, it's going to lead to high replication. And then you get lots of these cytotoxic T cells floating around in the blood, ready to kill the pathogens. Now, all is not always going to go so well when our body somehow, through different diseases and mechanisms, is going to lose the ability to recognize its own antigens. And depending on where that is going to cause different problems. So, for example, multiple sclerosis, MS, is when the body starts to mistakenly attack the myelin sheaths on our neurons when it's not supposed to, because that's a part of your own body. So an autoimmune disease is self-immune. It is targeting itself rather than things that are not itself. And for all of these different diseases, it's going to be attacking a different part of your body. So for example, type 1 juvenile diabetes mellitus, for that one, well, the body is now targeting the pancreas, specifically the beta cells of the pancreas, which is going to lead to an insulin deficiency. So whatever the autoimmune disease is, your body is now targeting itself. So if we have a hypersensitivity to some kind of foreign substance, we call that an allergy. So this is when we take something that's generally harmless and our bodies are having a reaction to it as though it is something harmful that needs to be taken care of rather than just passing through. So if we're talking about anaphylaxis, that would be, so you have your first exposure, there's no symptoms to whatever it is, let's say peanuts or something, not harmful, but our body might start treating it as harmful through the misapplication of the immune system. So then the second exposure, your mast cells, basophils, they're going to release histamine. You get this inflammatory response. It could be localized to that area, or it could be system-wide. You get reddened, itchy skin. We call that hives, watery eyes, runny nose. So that could come from you eating them, or maybe you inhaled them as some kind of particle. Maybe a certain tree blooms, it gets into the air and you breathe it in. Now, if you go into anaphylactic shock, that's going to be a life-threatening condition. And it depends on what you are going to have this reaction to. So it could be bee stings, spider bites. Some people are allergic to penicillin. So you could have your bronchioles constricting, vasodilation, and basically that could cause shock and then eventually death. And that's why we have these EpiPens. So the way an EpiPen works is epi stands for epinephrine. So it gives you a dose of epinephrine, and what that does is it relaxes the muscles in the airways. So that's going to allow you to breathe better, so you don't have this constriction of your bronchioles that could literally cause you to suffocate and have the shortness of breath. So the EpiPen alleviates that, which is one of the most important things that needs to happen. It's also going to increase your blood pressure, and that's going to allow you to move oxygen through your body faster, and that's going to help reverse the lower blood pressure that you get from anaphylactic shock through that vasodilation. So it's counteracting the vasodilation with vasoconstriction. It's going to reduce swelling. It's going to stimulate the heart, so the heart rate doesn't stop. So it actually increases the heart rate. And all those things are to keep oxygen and blood flow moving properly through your body so you don't have organ failure and you do not die. So EpiPens are super awesome. So if we say atopy, that means out of place. What that means is a genetic predisposition to developing an allergic reaction, which is going to cause you to overproduce immunoglobulins, or the antibodies, in response to things that are normally are harmless, and certain foods that are harmless. But our body, when we have atopy, we have this heightened risk for these certain diseases. So we might develop things like eczema or asthma and things of that sort. You can have delayed hypersensitivity, which would be, an example of this would be, contact dermatitis, like poison ivy. It's a delayed reaction, so it might take a day to 2-3 days before you see the reaction actually occur. So then we have what's called septic shock, or sepsis. So this would occur when the inflammatory response goes out of control, your blood pressure falls, body organs shut down, and then death will occur in half of the cases where septic shock happens. So example of that could be, bacteria all throughout your circulatory system. It's everywhere. That's going to cause a massive inflammatory response. Blood pressure is going to drop. Your body can't produce enough white blood cells to fight everything. And all of those systems go out of control to the point that your organs literally fail. And then you would die. And that concludes this part of the notes. I'm going to split this into two videos, because the second part, we're already going at 43 minutes. So I'll stop it here, and then we'll talk about the lymphatic system next.